Serious chronic diseases like diabetes and cardiovascular diseases are associated with chronic energy excess. One mechanism behind this is the nature of the response of the human adipose tissue to overnutrition. Imagine if new regulators of adipose tissue plasticity could be identified. This is what our CRISPR screening workflow aims to do. Brightfield imaging of adipocytes is a sensitive, scalable, and inexpensive readout. But how can regulators of adipose tissue plasticity be identified from brightfield images in a high-throughput workflow? Stay tuned to learn more. In a highly automated workflow, adipose progenitor cells are CRISPR perturbated and differentiated to adipocytes in multi-well plates. The wells are imaged with brightfield imaging, resulting in thousands of images where the cellular phenotypes such as lipid accumulation, cell size, and morphology of the adipocytes can be investigated, and where changes from the CRISPR perturbation can be revealed. Unlike staining that requires fixed cells, hence dead cells, and only captures endpoint phenotype, brightfield imaging can capture longitudinal measurement on live cells. We have trained the UNet deep neural network to generate virtually stained images from brightfield images, thereby enabling the readout of longitudinal data. How can we know if a CRISPR perturbation leads to a significant phenotype? We do this in 3 steps. First, we use Cell Profiler to convert an image into a vector of image features like lipid droplet count, lipid droplet intensity, granularity, and many more. Second, we train a support vector machine in feature space to distinguish positive regulators from non-targeting controls. In the third step, we calculate the distance to the SVM hyperplane of the positive and of the non-targeting controls. Based on the distances and to quantify how well our metric separates between the classes, we calculate our screening metric, the set prime value. The set prime value is a measure of separation of the sample distribution from the control. In our case, the positive regulators from the non-targeting controls. For a screen to have sufficient assay window, we'd like set prime to be positive. A set prime above 0.5 is considered excellent, as it allows for detection of very small effect sizes, almost regardless of sample size. PCA plots of feature space allows us to visually assess the separation of different classes of phenotypes in our assay into clusters. By shape coding different runs, we can observe that virtual staining is superior to fluorescent staining in removing batch effects and staining artifacts after normalizing plates to negative controls. A set prime of 0.33 is achieved for the separation of positive regulators of adipose tissue plasticity from non-targeting controls. This is a proof of concept that we can identify adipose tissue plasticity regulators from brightfield imaging with our workflow. Our workflow enables the screening for and the potential discovery of novel targets for improving human metabolic health. Thank you for listening.